Okay, so this is part 2 of chapter 20, the heart. Where we left off was we talked about the flow of blood through the heart. Now we're going to talk about the actual histology. If we look at the heart skeleton, it consists of a plate of fibrous connective tissue between the atria and the ventricles. These fibrous rings are around the valves for support. This is another layer of connective tissue really there to support this. The heart skeleton also serves as an electrical insulation between the atria and the ventricles as they are going to contract at separate times. The atria will contract first, the ventricles will contract second. It also provides a site for muscle attachments. Now, cardiac muscle tissue, you have elongated branching cells that contain 1 to 2 centrally located nuclei. They will contain actin and myosin myofilaments. It is a striated tissue. The sarcoplasmic reticulum will store calcium, but you do not have dilated cisternae that you oftentimes see with skeletal muscle. You have larger transverse tubules in close association with the sarcoplasmic reticulum, and this is found near the Z disc. Cardiac muscle is rich in mitochondria as you need to maintain ATP supplies for continuous contraction of the heart. You also have a very extensive capillary network, and calcium ions must diffuse a further distance from the sarcoplasmic reticulum to the actin myosin filaments. Some calcium comes from extracellular fluid and the T-tubules. Cardiac muscle tissue will also have intercalated discs, which are specialized cell-to-cell contacts. This allows cell membranes to interdigate. Desmosomes will hold the cells together, and the gap junctions will allow action potentials to move from one cell to the next. Electrically, cardiac muscle of the atria and the ventricles behaves as a single unit. You don't want part of your heart contracting and another part not contracting. So here you can see, this is sort of a summary from A&P1 where you sort of looked at cardiac muscle tissue. And again, here you can see the branching of the muscle cells, the intercalated disc, as well as the nucleus of the cardiac muscles, unlike skeletal muscle where you had multiple nuclei because you had several cells all fused together. Here, you just have individual muscle cells or fibers. Now, the conducting system, this is going to relay action potentials through the heart. Modified cardiac muscle cells that form 2 nodes at the conducting bundle are going to initiate the conducting system. These modified cardiac muscle cells are referred to as pacemaker cells, and there are 2 types. You have the SA node, which stands for sinoatrial node, and the AV node for atrioventricular node. The SA node is medial to the opening of the superior vena cava. This is the primary pacemaker of the heart. The SA node are specialized cardiac muscle cells that generate spontaneous action potentials. The action potentials pass to the atrial muscle cells and then to the AV node. Once passed to the AV node, this will end up firing the AV node. But let's talk about what the AV node is first. The AV node is located medial to the right atrioventricular valve. Action potential is conducted more slowly here than any other part of the system. And this ensures that ventricles receive signal to contract after the atria have contracted. So essentially, you're going to have the SA node fire, which will cause for the atria to contract, followed by the AV node firing, which will lead to the ventricles to contract. The AV bundle is going to pass through a hole in the fibrous skeleton to reach the interventricular septum. You will then have the right and left bundle branches, which extend beneath the endocardium of the apex of the right and left ventricles. And then finally, the Purkinje fibers. These are large diameter cardiac muscle cells with few myofibrils, many gap junctions, and their job is to conduct the action potential to the ventricular muscle. So right up here, this would be where the SA node is located. When this fires, this will then trigger the atria to contract. So the heart in this image is actually at rest and all chambers are at rest. The action potentials are generated at the SA node and spread from the SA node to the adjacent cardiac cells in the atria. Preferential pathways conduct action potentials, such as shown in the dotted arrows. From the SA node to the AV node at a greater velocity than they are transmitted in the remainder of the atrial muscle tissue. However, such pathways cannot be distinguished structurally from other areas of the atrial wall. What happens after the SA node fires is the atria contracts. The cardiac muscle in the atrial wall contracts in response to the action potentials conducted through the atrial wall. The action potential then passes from the AV node to the AV bundle. The bundle branches, and then the Purkinje fibers. And this will then stimulate ventricular walls to contract. When the heart beats under resting conditions, which is approximately 0.04 seconds, is required for the action potential to travel from the SA node to the AV node. The action potentials are propagated slowly through the AV node compared with the remainder of the conducting system. The slow rate of action potential conduction in the AV node is due in part to the smaller diameter muscle cells and fewer gap junctions in their intercalated disc. Like the other specialized conducting cells in the heart, they have fewer myofibrils than most cardiac muscle cells. As a consequence, a delay of 0.11 seconds occurs from the time the action potential reaches the AV node until it passes to the AV bundle. The delay in action potentials at the AV node allows for completion in the atrial contraction. So you want to make sure the atria contract and expel most of the blood into the ventricles. And this has to occur before ventricular contraction begins. After the action potentials pass from the AV node to the highly specialized conducting bundles, the velocity of the conduction increases dramatically. The action potentials will pass through the left and right bundle branches and through the individual Purkinje fibers that penetrate the myocardium of the ventricles. This will then cause the ventricles to contract from the apex towards the base. Because the arrangement of the conducting system in the ventricles, the first part of the ventricular myocardium that is stimulated is the inner wall of the ventricles near the apex. Thus, ventricular contraction begins at the apex and progresses throughout the ventricles towards the base of the heart. The spiral arrangement of muscle layers in the wall of the heart results in a ringing action. During the process, the distance between the apex and the base of the heart decreases and blood is forced upward from the apex toward the great vessels and the base of the heart. Like skeletal muscle cells, cardiac muscle cells have resting membrane potential and are capable of conducting action potentials. Resting membrane potential depends on low permeability of the plasma membrane to sodium and calcium, and a higher permeability to potassium. The heart is autorhythmic, unlike skeletal muscle, which must have nervous stimulation in order to contract. Autorhythmic means that it stimulates itself to contract at regular intervals. Pacemaker cells in the SA node generate action potential spontaneously. This is referred to as the pacemaker potential, spontaneously developing local potential due to movement of sodium, potassium, and calcium. And like you learned with skeletal muscles, once you hit threshold, the action potential is generated. So let's talk about the pacemaker potential. With the pacemaker potential, sodium ions cause depolarization by moving into the cells through specialized non-gated sodium channels. So these are not gated channels. They're essentially what we call leaky channels. The decreasing permeability to potassium also causes depolarization as less potassium moves out of the cells. The decreasing potassium permeability occurs due to voltage changes at the end of the previous action potential. As a result of depolarization, voltage-gated calcium channels will open, and the movement of calcium into the pacemaker cells causes further depolarization. Now, when the pacemaker potential reaches threshold, many voltage-gated calcium channels open. In pacemaker cells, the movement of calcium into the cells is primarily responsible for the depolarization phase of the action potential. This is different from other cardiac muscle cells, where the movement of sodium into the cells is primarily responsible for depolarization. Repolarization occurs as other cardiac muscle cells when voltage-gated calcium channels close and voltage-gated potassium channels open. After resting membrane potential is reestablished, production of another pacemaker potential starts the generation of the next action potential, and you start the whole cycle over again. Now let's look at the action potentials in the cardiac muscle, and you'll note that this figure looks a little bit different than what you saw with the action potentials in skeletal muscle. The first phase is the depolarization phase. The rapid depolarization is a result of changes in membrane permeability to sodium, potassium, and calcium. However, membrane permeability to sodium is the primary determinant of the phase. Membrane channels called voltage-gated sodium channels open, bringing the depolarization phase of the action potential. As a result, voltage-gated sodium channels will then open. Sodium diffuses into the cell, causing the rapid depolarization until the cell is depolarized to approximately 20 millivolts. Now, even though you have the membrane channels opening in response to a change in voltage for sodium, you get that huge influx. The voltage change occurring during depolarization will affect other ion channels in the plasma membrane as well. Several types of voltage-gated potassium channels exist, each of which opens and closes at different membrane potentials, causing changes in membrane permeability to potassium. So for example, at rest, the movement of potassium through the open voltage-gated potassium channels is primarily responsible for establishing the resting membrane potential in cardiac muscle cells. This depolarization causes these voltage-gated potassium channels to close, thereby decreasing membrane permeability to potassium. The depolarization also causes voltage-gated calcium channels to begin to open. These channels contribute to depolarization. Compared with sodium channels, the calcium channels open and close relatively slowly. During the early repolarization phase, now this is where you're going to see some difference to that of skeletal muscles. Early repolarization phase, repolarization is also the result of changes in membrane permeability to sodium, potassium, and calcium. Early repolarization occurs when voltage-gated sodium channels and some voltage-gated calcium channels close, and a small number of voltage-gated potassium channels open. Sodium ion movement into the cell slows, and some potassium moves out of the cell. At this point, repolarization begins, but in cardiac muscle, early repolarization is slow due to the influx of calcium, resulting in the plateau phase. Now, the plateau phase occurs as voltage-gated calcium channels remain open, and calcium and some sodium move into the cell through voltage-gated calcium channels. The influx of these ions counteracts the potential change produced by the movement of potassium out of the cell. Finally, the repolarization phase. The plateau phase ends and the final repolarization begins as the voltage-gated calcium channels close and many more voltage-gated potassium channels open. Thus, calcium and sodium stop diffusing into the cell, and the tendency for potassium to diffuse out of the cell increases. These permeability changes cause the membrane potential to return to its resting levels. Now let's kind of go back and cycle back, now that we've talked about the action potential, on some of the differences between skeletal and cardiac muscle physiology. With cardiac muscle, action potentials are conducted from cell to cell, whereas in skeletal, action potential is conducted along a length of a single fiber. With cardiac, the rate of the exponential progression is slow because of gap junctions and a small diameter of fibers, whereas it's much faster in skeletal muscle. Cardiac muscle has a prolonged period of slow repolarization called the plateau phase. Skeletal muscle has a rapid repolarization with no plateau. Cardiac muscle, you have calcium-induced calcium release, which is the movement of calcium through the plasma membrane and T-tubules into the sarcoplasm will stimulate the release of calcium from the SR. Skeletal muscle doesn't depend on this mechanism. It relies mostly on intracellular calcium for concentration. And here you can see the comparison between skeletal muscle and cardiac muscle as far as the action potential is concerned. Now, the refractory periods of cardiac muscle. The absolute refractory period is when cardiac muscle cell completely is insensitive to further stimulation, meaning no contraction could occur during this phase. The relative refractory period, this is when the cell exhibits reduced sensitivity to the additional stimulus, meaning that it's probably going to not contract during this period, but it could if it needed to. All right. Shifting gears a little bit here, let's talk about an ECG or an electrocardiogram. An electrocardiogram is a record of electrical events in the myocardium and can be correlated with mechanical events. You have what we refer to as, this is a typical EKG. You have the P wave, the QRS complex, and then the T wave. During the P wave, you have depolarization of the atrium myocardium and signals onset of atrial contraction. So what's happening during the P wave is atrial depolarization, which means the atria are contracting. During the QRS complex, you have ventricular depolarization and signals onset of ventricular contraction. So during the QRS complex, you are having ventricular depolarization, which means the ventricles are contracting. Now, because the atria are repolarizing during the same time as ventricular contraction is going to mask the electrical activity that we would typically see on an EKG, and you're not going to see it. But the atria are also repolarizing at the same time as the QRS complex. The T wave is the repolarization event of the ventricles. This precedes ventricular relaxation. Your PQ interval, or the PR interval, is the atria contract and begin to relax, and the ventricles begin to contract. The QT interval is when the ventricles contract and begin to relax. Okay. All right. There are many different types of arrhythmias, or what we refer to as irregular heartbeats. And you can have tachycardia, atrial flutter, or atrial fluctuation, atrial fibrillation, and ventricular fibrillation. And you can sort of look at some of the symptoms that you typically see and some of the causes that this could be a result of. Okay. Okay, so now we're going to talk about the cardiac cycle. Now, the heart, as I mentioned earlier on, is two pumps that work together. You have the right and left halves. Each pump has a primer pump, the atria, and a power pump, which are the ventricles. Now, the cardiac cycle is a repetitive pumping process that begins with the onset of the contraction and ends with the beginning of the next contraction. Systole is contraction of a chamber. Diastole is relaxation of a chamber. Blood moves through the circulatory system from areas of higher to lower pressure. Contraction of the heart produces the pressure. So the first phase of the cardiac cycle is the heart is relaxed. So you have passive ventricular filling. As stated earlier, before the cardiac cycle begins, all chambers are relaxed. At rest, most of the blood movement into the chambers is a passive process, resulting from greater blood pressure in the veins than in the heart chambers. As a result, blood moves into the atria, and much of it flows into the ventricle for two reasons. One, the AV valves are open, and two, the atrial pressure is slightly greater than ventricular pressure. This time period, when blood is passively moving into the ventricles, is called passive ventricular filling. Atrial systole, this is active ventricular filling. This is when the SA node generates an action potential and stimulates the atria contraction. The P wave of the ECG represents this electrical activity. Atrial contraction begins the cardiac cycle. As the atria contract, they carry out the primer pump functions by actively forcing more blood or any remaining blood in the atria into the ventricles. Now, ventricular systole, this is the period of isovolumetric contraction. The action potential passes to the AV node, down the AV bundle, through the bundle branches, and into the Purkinje fibers. This will trigger ventricular systole. This electrical activity is represented as the QRS complex of the ECG. As the ventricles contract, ventricular pressures increase, causing blood flow towards the atria and close the AV valves. Recall that the semilunar valves are closed at this point as well. Ventricular contraction continues and the ventricular pressure rises. However, because all the valves are closed, no blood flows from the ventricles at this time. This brief interval is called the period of isovolumetric contraction because the volume of blood in the ventricles does not change, even though the ventricles are contracting. The fourth phase of the cardiac cycle is ventricular systole, the period of ejection. The ventricle contraction continues, and ventricular pressure builds until it overcomes the pressure in the pulmonary trunk and the pressure in the aorta. As a result, the semilunar valves are pushed open, and blood flows from the ventricles and into those arteries. The fifth phase is ventricular diastole. This is the period of isovolumetric relaxation. Ventricular repolarization is represented by the T wave of an ECG. This leads to ventricular diastole. As ventricular diastole begins, the ventricles relax and the ventricular pressures decrease below the pressures in the pulmonary trunk and aorta. Consequently, blood begins to flow back towards the ventricles, causing the semilunar valves to close. With closure of the semilunar valves, all the heart valves are closed and no blood flows into the relaxing ventricles during the period of isovolumetric relaxation. And here's those 5 steps sort of summarized up again. Atrial systole and active ventricular filling. Let's cycle back on one of the concepts here. The depolarization of the S8 node generates action potentials that spread over the atria, again, the P wave. The atria contract, and this completes ventricular filling. At rest, the contraction of the atria is not necessary for heart function. During exercise, atrial contraction is necessary for function as the heart pumps 300 to 400% more blood than during at rest. So essentially what this is saying is that the normal passive movement of blood from the atria to the ventricles, even without atrial contraction, is sufficient to amount of blood to be pumped out through the ventricles. Ventricular systole is the period of isovolumetric contraction. Again, this begins at the completion of the QRS complex. The ventricle muscles start to contract, increasing the pressure inside the ventricles. This causes the AV valves to close, which is the beginning of ventricular systole. The semilunar valves were closed in the previous diastole and remain closed during this event. Approximately 100 to 130 mils of blood are in the ventricles, left from the last diastole when the atria emptied into the ventricles. This is referred to as the end diastolic volume. The period of ejection. the pressure in the ventricles has increased to the point where it is greater than the pressure in the pulmonary trunk and the pressure within the aorta. This now allows for pushing the cusp of the semilunar valves against the walls of the vessels, opening the valves. Blood is ejected from the ventricles. The pressures in the two ventricles are different, 120 in the left ventricle, whereas it's only 25 in the right ventricle. This is because on the left side, you have to pump blood through the entire systemic circulation, meaning the blood needs to get from the heart to the toes and back from the toes up to the heart. On the right side, you're only pumping blood through the pulmonary circulation, which means blood only needs to get from the heart to the lungs. And remember that the heart and lungs are literally right next to each other, so you don't need high pressure to get that get through that loop. After the first initial spurt, the pressure starts to drop. At the end of the period of ejection, 50 to 60 mils remain, and this is referred to as the end systolic volume. Now, the period of isovolumetric relaxation, the completion of the T waves results in ventricular repolarization and relaxation. The ventricular pressures fall very rapidly. The pulmonary trunk and aorta is higher than ventricular pressure. You then have an elastic recoil of the arteries, causes blood flow back towards the relaxed ventricles. At this point, the semilunar valves close, which is the beginning of ventricular diastole. Note that the AV valves are also closed. While the ventricles were in systole, the atria were filling with blood. Atrial pressure rises above ventricular pressure and the AV valves open, and blood flows into the relaxed ventricles, accounting for most of ventricular filling. About 70% of the blood will flow passively from the atria into the ventricles. Here's a figure that sort of summarizes the electrical activity, the EKG. Here we have the pressure flowing through the various structures. Here is the left volumetric volume of blood, and then our heart sounds. The heart sounds are a result of the opening and closing of the valves. So the first heart sound, or the lub, is when the atrioventricular valves and surrounding fluid vibrations as the valves close at the beginning of ventricular systole. The second heart sound, of the dub, results from the closure of the aortic and pulmonary semilunar valves at the beginning of ventricular diastole and lasts a little bit longer. You can occasionally hear a third heart sound. This is caused by the turbulent blood flow into the ventricles, and it's detected near the end of the first 1/3 of diastole. Abnormal heart sounds are referred to as heart murmurs. These could be either a result of an incomplete valve formation or as a result of a stenoic valve. A stenoic valve means that it is sort of hardened up a little bit and it's not as flexible as it used to be. When you go for a physical, your doctor will listen to your heart and usually will move the stethoscope over the chest to 4 specific areas. And the 4 areas are indicated here by the little red dots. And this is really just so that they can hear each of the 4 valves. Okay, looking at an aortic pressure curve, you have what's referred to as a dicrotic notch. This is when the aortic semilunar valve closes. This is actually going to cause the pressure within the aorta to increase slightly. So it's sort of like blood flows out, kind of hits the wall, comes back, closes it to close, and that sort of increases the pressure a little bit. All right. Okay, so now we're going to talk about mean arterial blood pressure, or MAP. This is slightly less than average blood pressure in the aorta. The mean arterial pressure is the cardiac output, which is the amount of blood pumped by the heart per minute, times the peripheral resistance, which is the total resistance against which blood must be pumped, meaning the pressure that has to be achieved so that blood flow will move. There are several factors that can affect mean arterial pressure. Oops, sorry. And those are sort of listed here, such as either increasing blood volume or decreasing blood pressure. These are all going to have effects on the overall mean arterial pressure. Now, regulation of the heart output must change to meet the demands of the body. You have intrinsic regulation, which results from normal heart function, whereas extrinsic regulation involves neural or hormonal control. With intrinsic regulation, preload is the amount of stretch on the ventricular walls. The greater the stretch, or the greater the preload, the greater the force of the contraction. This is known as Starling's law of the heart. Afterload is the pressure the contracting ventricles must produce to overcome the pressure in the aorta and move blood into the aorta. Heart not as sensitive to this as it is to changes in the preload. With your extrinsic regulations, this will involve both neural and hormonal control. The parasympathetic stimulation is going to supply by the vagus nerve. Decreased heart rate, acetylcholine is secreted and hyperpolarizes the heart. Sympathetic stimulation is going to be supplied by the cardiac nerves. This will innervate the SA and AV nodes, the coronary vessels, and the atrial and ventricular myocardium. This increases heart rate and force of contraction. Epinephrine and norepinephrine are released. Increased heartbeat causes increased cardiac output. Increased force of contraction causes a lower end systolic volume. The heart empties to a greater extent. Hormonal control, epinephrine and norepinephrine from the adrenal medulla. This occurs in response to increased physical activity, emotional excitement, stress as well. This oftentimes will take longer than sympathetic stimulation, but it does last a bit longer. Now, the effect of blood pressure. Baroreceptors will monitor blood pressure in the walls of the internal carotids and the aorta. This sensory information goes to the cardioregulatory system of the brain in the medulla, which can then increase or decrease the heart rate. pH, carbon dioxide, and oxygen can also all have effects on heart rate as well. These chemoreceptors are going to cause what we call chemoreceptor reflexes, where you can then see alterations in that. Okay. Sorry, just kind of, this will go over some of the chemoreceptors and baroreceptor reflexes. Body temperature also will have an effect on heart rate. When your body temperature increases, this will cause the heart rate to increase. And when body temperature decreases, this will also cause the heart rate to decrease. Age-related changes are minor under resting conditions, but are more significant with exercise and other age-related diseases. Age-related changes of the heart can include hypertrophy of the left ventricle, increased pressure in the aorta, lipid collection on the myocardial cells, maximal heart rate declines, hormone effects decline, valves become less flexible, congestive heart disease, and coronary artery diseases. And here are some of those conditions and their descriptions. You can kind of look through these. I don't specifically necessarily test on these, but they're just up here for your information. This is a good figure because this actually shows you what happens with a myocardial infarct, which is a heart attack, and how it affects almost every system in the body. So this is also an interesting little read. Okay, and that is it for the heart.